Hi, so in this video I'll be talking about some of the areas that you might want to concentrate on with your upcoming Unit 4 exams. This video is directed towards the 2026 May-June Cambridge exams Unit 4. So let's do a deep dive in and let's see what you probably won't want to study before the exam is on. So this is not a prediction for the exam, but this is basically the analysis of the 2026 February-March paper and also the 2025 papers and the ones before. So we are going to look into areas that has not been tested in the recent papers and area. So if you look into the syllabus, one of the first lessons that we have is about energy and respiration. So in the recent papers, there was a question about glycolysis and there were some questions about the electron transport chain as well. Nevertheless, you need to know what respiration is all about, and you also need to know why ATP makes it a good universal currency as well, because these are areas that has not been tested, especially about why ATP is considered as a universal energy currency. So the syllabus itself tells you why it is a good energy currency. So you might want to go through that. And it also talks about how ATP is basically being synthesized in both mitochondria and chloroplast. Now we know that ATP is where the energy is stored, and we always relate this with respiration. But we sometimes do forget that there is ATP production even inside the chloroplast as well. So you could expect a question where both respiration and photosynthesis is taken into one single question with the help of this ATP. So you might want to consider going through how phosphorylation takes place in both these processes. Another question that you might want to look at is questions on the respiratory quotient. Now, first of all, you should know what respiratory quotient is, and there are two ways you can calculate respiratory quotient. One way which we normally do is to find the ratio of the volume of carbon dioxide given out over the volume of oxygen that was used up. Now, you would also want to go through the respiratory quotient values for carbohydrates, proteins and lipids, and all these values are given in your reference books, so you should know them by heart. Now, one of the other things that I have highlighted over here related to respiratory quotient is the use of a respirometer to calculate the respiratory quotient. Now, this is something that could appear in your Unit 5 as well, because it's something to do with planning and experiment as well. But nevertheless, you still might want to go through the experiment about the respirometers, how you can measure the volume of oxygen from this, right? It'll most probably appear in Unit 5, but then again, you still might want to go through the theory of this as well. Then in terms of the process of respiration, so as I said, in very recent papers, there had been question on glycolysis. There's a very interesting question very recently where they gave the inner membrane of the mitochondria, where the children had to identify where NAD and FAD go and dump their H+ ions. Now, one of the things that has not been tested in terms of aerobic respiration is the Krebs cycle. Now, Krebs cycle, there is so much you can be tested on, and one of the most common things that we see in past papers is a fill in the blank. So you should know the entire process, what is being produced during each process. So something you really want to go through in terms of theory, right? Another thing would be the oxidative phosphorylation still, because as I said in the previous questions, the recent questions, they mainly talked about the process of electron transport system, but still there could be a good question that could appear in terms of how ATP synthase gets involved in the ATP production. This relates to what I said before at the beginning of this video, where you might want to go through how ATP is being produced in photosynthesis as well as in respiration as well as why ATP is still a good universal currency as well. Then passing through, though it sounds very simple, you might want to also go through a summary about the number of ATP, the number of NADP, the number of NAD and the number of FAD that are being produced during respiration. Because you could always get a table where you need to summarize these things as well, right? So you might want to know the amount of these products that are being formed. So as I said, Krebs cycle is a favorite for this time because it has not been tested for a long period of time. That's one of the reasons why I've highlighted everything that is to do with the Krebs cycle over here. Then moving further in terms of respiration, one of the recent questions talked about, I think this was the recent February-March paper, how the paddy plants are adapted for waterlogged soils. And this is again something to do with respiration. But I have still a feeling that you probably might get a question where you're given a photomicrograph of Ehrenheimer, right? Which is one of the structural adaptations of a rice plant to make sure that it can store air and how it can transport the air as well to the roots that are underwater, which are submerged underwater. So this is where you probably might want to be thorough with your theory once again, even though it came in one of the recent papers, because this would be a very good photomicrograph for the children to identify, because it also has a cross-section of a stem as well. Now, if you go through your syllabus, this is something which most children do not know. Your syllabus itself says that when it comes to your unit 4 papers, this is not just going to be about your AS knowledge. As you can see, they have also said knowledge of material from the AS level syllabus content also will be required, which means a structure or a cross-section of a stem, you probably would need to know how to label it as well. Okay. Going a little bit further again into respiration, one of the areas you might want to also be thorough with is the experiment on DCPIP. Again, this could be a unit 5 area, but you can still get a very small question about asking about the DCPIP or methylene blue. How does it basically work in terms of respiration? What can you measure out of it, right? So you probably might want to be thorough with the theory of that as well. So this is what I would say you really want to concentrate in terms of respiration. And let's now move on to photosynthesis as well. Now, even though this sounds very easy, you still probably would want to be thorough with a good definition that you can write for photosynthesis. Now, this means you should not be writing the same definition that you wrote when you were doing your GCSEs. So you would have to talk about the organic products that are being formed. You might want to give reference to light energy, right? And you might want to talk about photosynthetic pigments. But it has to be a good definition that shows that you are as an A-level student. Another thing that you would want to concentrate is to describe the relationship between the structure of chloroplast, and this can be related to an electron micrograph once more. Now this means that you probably could get a picture of a photomicrograph, that means a picture under the microscope of a chloroplast, where you have to label the parts, and they could ask you, where does the light dependent reaction happen in this picture, or where does the light independent reaction happen in this picture. And one of those questions where you may have to write a bit, like a 5 mark or a 6 marker, they could ask you about the description of the structure of the chloroplast in terms of its function. So you need to know about each and every stage in photosynthesis. Where does it happen? And you want to do a deep dive about the proteins that are involved, the enzymes that are involved, and you can even talk about how the structure helps to maintain the concentration gradient for the production of ATP and stuff, right? So this is something I feel that you might want to prepare for, just in case that you get an essay question on this part. Now, there had been some recent questions again on photosynthesis, and one of the most recent papers had a question about the absorption spectrum as well as the action spectrum at the same time for chlorophyll. Now, this does not mean that you will not be tested on this part, but obviously you'll need to go through this part once more, but you would really want to be thorough about the use of this knowledge in agriculture. Like, for example, what can we do after learning about the action spectrum? How do we use it now in day-to-day life? So that's something that you might want to read about. You may have heard about growth lights, which uses the blue color, red, and even a bit of violet, which helps in photosynthesis. So a question like that, an extended question like that, is possible for you this time. So that's something that you might want to go through. Another area that have not been tested in the unit 4 is about the chromatography. Again, chromatography could appear in your unit 5 because it's a practical oriented question, but nevertheless you have a lot of theory involved with chromatography as well, because chromatography helps you to identify different pigments. You would be asked to calculate the RF values, which means you need to know the equation to calculate the RF values as well. Now, they could give you a table with standard RF values, and they could basically ask you to calculate your RF value and try to find out what pigment is being shown in your chromatographic paper. So that's something you really want to be aware of. In terms of photosynthesis, again, a question on cyclic photophosphorylation and non-cyclic photophosphorylation. Now, this means that you could be given a picture of the thylakoid membrane, and you probably might get to label the parts that are involved in the cyclic and non-cyclic photophosphorylation. Now, if they were to combine this, they probably might ask you to identify the parts first and then ask you what are the parts that are not found in the in the cyclic photophosphorylation. So this is something again you might want to go through, and you should be able to describe the process as well. Then again, coming back to the cyclic non-cyclic photophosphorylation, you should ask yourself, why would there be a cyclic photophosphorylation when non-cyclic photophosphorylation could also happen? So you might want to ask yourself, and you might want to research a little bit, when would the plant shift between non-cyclic and cyclic photophosphorylation, just in case that this do come as an extended question to you. And you also should be able to talk about the differences between these two types of respirations as well, as in what is the difference between cyclic photophosphorylation and non-cyclic photophosphorylation, because we have not seen this kind of a question again for a very long time. Then going forth, you would want to go through the stages of the Calvin cycle. You should be able to talk about what happens in the 3 main stages of this Calvin cycle, the products that are formed, and you should be able to describe it as well. Now, I have a feeling, just like the Krebs cycle that I mentioned, they could always give you a picture of the Calvin cycle and ask you to fill in the blanks with some of the products, some of the enzymes involved in that reaction, right? So that's something again you might want to go through. Now, another area that has not been tested for a while is about the limiting factors in photosynthesis. Now you can see I've highlighted all the 4 main points mentioned in this syllabus, because one thing is you need to know what these limiting factors are, And one of the major mistakes children do is that when they are asked for a limiting factor, they just go and say carbon dioxide. Now, carbon dioxide doesn't say that it's a limiting factor. You need to say that it's the concentration of the carbon dioxide that becomes the limiting factor. So your wording needs to be specific, precise, especially when you're answering the Unit 4 papers. Another thing that you might want to consider here are the graphs that you have learned in your theory. How does carbon dioxide would affect as a limiting factor? Because you might remember how the limiting factor graphs would be, and you have done these in your GCSE as well, which means you need to be very thorough with that now. Another area is again about the use of DCPIP or methylene blue to analyze the redox reaction that is happening during this process as well. But as I said, this is a more experiment oriented question, which means you would expect this more in your Unit 5, but then again you need to be sure about this theory as well. Another thing over here is the description of how to use an aquatic plant to measure the rate of photosynthesis. Again, this could be both a theory in our Unit 4 as well as a more towards Unit 5, because it's again an experimental question. But you still might want to be sure about the theory just in case there may be a question on this part as well. So coming to homeostasis, very interestingly, in the more recent papers, there had been more questions towards homeostasis. And one of the most recent questions that we saw was a question about the nephron. And there was also a question about the lumen of the nephron and the cells that are there in the nephron as well. Now, one of the things that they have not given as a question for a very long time is to observe a structure of a kidney. Like they give you a cross section of a kidney and ask you to identify the parts that are mentioned over here. Now, this kidney could be a practical question once more, or they could actually give you a real picture of a cross section of a kidney and ask you to identify the parts in it. So that's something you might want to go through as well, because they could always ask you the location of the different parts of the nephron in that kidney. They could ask you to draw a model nephron into the structure of the kidney just to see where certain parts of the nephron are found. Going forth, you can also be tested about again an actual photomicrograph of a nephron. Now this is something that is very challenging to identify things like the proximal convoluted tubule and the distal convoluted tubule. Now there's a very good picture given in your textbook or your reference book, actual photomicrograph and a hand-drawn picture of the different parts labeled as well. So you might want to train your eyes how to find out what each of these sections of the nephron are if it is given as a photomicrograph. This means if you do get a question like that, they could always ask you about the formation of urine. That means to talk about ultrafiltration, to talk about the selective reabsorption, because that is again a very good extended question that can be related with both the kidney as well as your nephron as well. We've also seen a very recent question about ADH, but I still feel that ADH as a question is still possible because there's so much you can test on this. This is basically something that you can relate to coordination, how the osmoreceptors basically detect the water potential and how this coordinates with the pituitary gland to release ADH. And then in terms of homeostasis, how does it basically help to balance the water levels in the body? So this is one question that you still might want to be sure about the theory, even though there had been a lot of questions in the recent past. Going further, there is an area about cell signaling, but in a very recent paper there was a lot about cell signaling, but I'm not saying not to go through it. As I said, this is not a recommended one just to say that these are the only things coming for the exam. but these are areas that we are looking and analyzing. What you really want to concentrate at this last moment now, just run through the syllabus behind cell signaling, but I have a feeling that you might really want to concentrate on this part over here, where it says explain the principles of operation of test strips and biosensors. Now, already there was a small question regarding a test strip in a recent paper, but regarding the biosensors and the enzymes that are involved. Now, when I say enzymes here, you know that this is very much related to the lesson on enzymes again, and this talks about enzymes that are stuck on one place, that cannot move around. These are the immovable enzymes, right? So you might want to go through the theory behind this, and your textbook does talk about, and even your syllabus does talk about, what these enzymes do. Like, how does it break one certain product, the glucose, into another product? How does the color change happen, indicating the sugar levels to you? So you might want to really go through this in detail, and because I feel that there is a question that probably could come from this section, as this has not been tested again for a very long time. So moving further, very interestingly again, there has been a lot of questions related to plant homeostasis, especially when it comes to the hormones in plants. And in a very recent question, there was something to do with some of the hormones that would be controlling the growth of the plant stem, and that was actually connected even with the previous one which I was talking about, the rice plants. How does the rice plants grow when it is subjected to a waterlogged soil? So in terms of homeostasis, I feel this time it will be more directed towards the human one, which you might want to concentrate more on. But yes, obviously you need to go through the plant one as well. Moving even further to the coordination lesson. Now, there had been a mixture of questions from coordination. Even in the 2025 papers, there was a nice balance between both plant coordination and animal or human coordination as well. But some of the areas that have not come again for a very long time is about the identification of different neurons, where they could give you an actual picture of a motor neuron or a sensory neuron and ask you to identify them, and also ask you to talk about its functioning. How does it basically transport its information from one end to the other? And you could see over here they've talked about intermediate neurons but not spoke anything about the structure. But when it comes to the sensory and motor, they have been talking about both structure and function. So a photomicrograph, an image of this is something possible, and you need to be able to talk about the differences between the two, right? So go through the structural differences in these two cells, because these are an area that you might want to be thorough as well. Another area that you probably might want to be sure about is the graphs that you see with related to the resting to action potential and the refractory period. They could always ask you about the movement of ions that takes place in each of those stages during a nerve impulse transmission. So that's something that has not been tested again for a long time. So you might want to be thorough about that graph, what happens at each section, and how the ion movement happens across the membrane as well. Another area that was slightly tested in one of the recent papers was about how acetylcholine causes the calcium ions to be released in striated muscles to go and join to troponin. Now, that was one big question which related coordination with the synaptic part, and they also related that with the muscular contraction as well. Now, in terms of theory, what should you now concentrate about is again obviously about the synapse, like what are the steps involved during the synaptic transmission. You should be able to list out the steps one by one, and you should know about some of the neurotransmitters as well. Now, another way you can be tested here is a cross-section of a striated muscle. That means they can actually give you a striated muscle, ask you to identify the cells in it, and they could ask you about the parts of a muscle fiber, where they can ask you to label the T-tubules and all that. So you might want to train your eyes again in this, and you might want to go through your notes or your textbook on the notes about this particular section. Another area you might want to still go through again is the sliding filament model. Now, yes, there had been a few questions about troponin and tropomyosin, but I mean, they've just slightly touched on the theory, but they've not gone into the details of asking you about the sliding filament theory once more. And also, I just mentioned you about the ultrastructure of the striated muscle. You can see this in the syllabus as well. It says that you should be able to describe that structure, and obviously you should be able to identify the parts if given under a photomicrograph, and this is something we've not seen at all. Going a little bit further on this, so Venus flytrap, there was a big question on it in 2025 paper. Now this does not mean that it won't come, but the probability is very less in this. You might want to still go through a little bit about, you know, its trapping mechanism and all that, and that was exactly what was tested in 2025 anyway. Going on to inheritance, now if you look into some of the past papers in the recent years, the 10 questions have basically covered up all the lessons in the A2. So there is always one question at least from every section, and inheritance is obviously a question that will always come in your paper. Now, some of the things that you might want to go through once more are the definitions that they have spoken about in this lesson. You need to be thorough with those definitions, because almost every paper or every variant paper, there is some sort of a question when it comes to the definitions of what is a gene and allele. I mean, they sound very easy, but you want to find out whether you really know how to write it down. Another area that you might want to go through are the stages of meiosis. Now, even in a recent paper, there was a photomicrograph of a few stages of meiosis asking about what happens and all that. But then again, you still need to be very thorough with this section, because when it comes to meiosis, there are so much of differences when compared to the mitosis, especially in meiosis 1. And as I said before in this video, even though you're doing your Unit 4 paper, you still need to be thorough with your AS work as well. So they could always ask you to compare between meiosis and a particular stage in, a stage in meiosis 1 or 2, and compare that with mitosis as well. So again, you need to be really, really thorough with this theory over here. So as I said, this 4 and 5 talks about photomicrographs and the stages and the differences of what happens. And you should also be able to compare between meiosis 1, a particular stage, with the same stage in meiosis 2 as well, right? Then another thing that you might want to be thorough with is this number 6 and number 7. How does meiosis help to create genetic variation? Because you know that genetic variation is really important when it comes to bringing about evolution, bringing about variation that helps organisms to adapt into the environment. So you should be able to describe this process in much detail, which would basically help you out. Another thing that you might want to go through, as I said, is the definitions that I have highlighted as number 1. You should basically know all these definitions, what they are, and you can also be given questions on this part. Now, an area that we have not seen in the Unit 4 paper is questions on codominance, questions on gene linkage, right? And even a dihybrid cross is something, that's what I have highlighted number 3 over here. A dihybrid cross and a autosomal linkage question. Now that is a little bit different from the standard crossing that you do, because in the case of a gene linkage, as you might know, the alleles do not separate. So especially when it's come to a dihybrid cross, you see both the alleles going together into the gametes, which means the number of variation in the gametes will be limited. So that's something that you might want to be aware of, because you would obviously need to know the monohybrid crosses and the dihybrid crosses, what are the phenotypic ratios. Because when it comes to gene linkage, these ratios will not be the same as a normal dihybrid cross or a monohybrid cross. So you might want to try a few questions. Your textbook has good questions on this, and even the exam style questions, there are good linkage questions, as well as there's one question on epistasis as well. Now again, epistasis, you don't need to know the knowledge about how it happens and all that, but they could always give you a question saying that, you know, there is a particular gene in a particular organism, and that gene has an effect on the other gene which you want to look the inheritance on. So there are some good examples again in your textbook which you might want to go through and be aware about epistasis, because we have not seen epistasis, neither gene linkage, for a very long time. So that's a very challenging question, right, which you might want to go through. Again, test crosses is something which has not been tested again, so you need to know what a test cross is, and you should know why do we do a test cross, and how to do it as well, and what ratios you can expect using a test cross. Then again, chi-square is something that you can be again tested. It's better if you know the formula of the chi-square. So you probably would not get a big question where you need to do the chi-square calculation on your own. That is very unlikely because it's time consuming, but you still could get some fill in the blanks in a table of chi-square, and then calculate the chi-square value. Now here you would obviously get a standard chi-square table. This is a probability table in chi-square, and you might want to compare your calculated chi-square value with the chi-square value that is given on the table. So this is something I feel that you might want to practice before this exam paper that is coming up, because we have not seen chi-square for a long time. Then coming to 16.2, you could see over here that I have highlighted the whole of number 6 over here. Now, one of the most recent questions we saw was one on Huntington's disease, where they actually asked the children the name of the gene that causes Huntington's disease. Now, that is something that came after a very long time, where the children had to show that they knew the exact gene behind each of these diseases. This means you might want to brush through the others as well, because Huntington's was already tested. Now, this doesn't mean that Huntington's won't come, but you still would want to go through the rest of it as well. Also, there was a very recent question about the gibberellin synthesis and how it affects. So I really doubt that they would want to test that again in this paper, but again, you still would want to go through the theory. Gene control is something, again, you can just brush through on, but because there was a very big question on an operon, about how the lac operon is basically involved, and they also basically related that with the gene controlling as well. So I would say you might want to just brush through on that theory a little bit. Then when it comes to selection and evolution, again, there was an interesting question about evolution in a very recent paper. This was in a 2025 October, November, I believe. Nevertheless, you still would want to know what variation is, and you should be able to talk about both continuous and discontinuous variation. I know it sounds easy, but you want to ask yourself, do you know how to define this? And do you know any examples that you can tell about both of these types of variations? And what causes these variations as well? Now, if you're given some data regarding variation, you probably might get a t-testing, right? You might be given the formula there, find the t-test value, or they would give you the t-test value, and through a probability table, you might want to say whether there is a significant difference or not. Again, this is more towards the unit 5 in terms of theory, but you still would want to be thorough with that. In terms of natural selection, Again, as I said, there was a good question about natural selection very recently, but this does not mean that it won't be tested, because they could always ask you the process of natural selection, like they give you a particular story behind a particular organism that was there a long time ago, but now you see a slightly different organism. How did this happen? And you know that there is a script that we always write when it comes to natural selection, right? Just compare some past paper questions on natural selection and you will see that it's the same script that goes over and over again. So you might want to be sure about what you're going to write over there. Very interestingly, once more, there was no question about this section over here, about asking you about the genetic drift. I mean, there was a little bit of a theory that they asked, but not a very big question about the founder's effect, and how a new population starts stabilizing in a new environment. So that's something you might want to go through, because that talks about speciation as well. Now, another area we have not seen in the Unit 4 papers in the most recent February, March, neither in the 2025 paper series, is about it's about Hardy-Weinberg's theory. Now, this is something I feel that you could get a big question where they might give you some values, ask you to calculate the frequency of them, and then use the Hardy-Weinberg's equation to do an estimate about maybe the heterozygosity of it and all that. So you would want to go through this, and you might want to practice a few questions. As I said, your textbook has really good questions on Hardy-Weinberg's theory. And there are lots of YouTube channel videos that you might want to refer to that talks about Hardy-Weinberg's equation and some practice questions on it. So please do practice some questions, especially in ones where you're given the numbers of the organisms and you need to convert them into a frequency and then use the equation to find those values. So that's something you might want to practice again on. Selective breeding is something that has been popping up in the very recent papers, and there have been quite a lot of questions on selective breeding, which means you might want to still be aware of selective breeding, especially when it comes to describing the process of selective breeding, like how do you do the selective breeding and all that. Now, again, this follows a particular script, right? You take some past papers, you go through it, and you see it's the same points, it's the same order that they give marks for. So you might want to practice a little questions on that part. And here are some examples given in your syllabus for selective breeding as well, which obviously you might want to go through, because there was one question about rice very recently. So you might want to go through the others in the syllabus. Then coming to evolution, yes, there was a question on evolution in the recent papers, both February, March, the May, June and October, November series. Even the variant papers had these questions on evolution. But then again, I think that they have not tested much about allopatric speciation and the sympatric speciation. So you might want to know what they are and try to explain based on a particular question. Maybe like they give you about a particular organism with some data about the history of the organism in a particular island, and try to use your knowledge about these two types of speciation and try to relate it as well. So that's something you might want to check on. Then when it comes to classification, again, very interestingly, there has been very low questions in terms of theory about the three different types of the domains, like the archaea, the bacteria and the eukarya. Their features. You know, give an organism and ask you to identify what type of domain that organism belongs to, right? And archaea is one of those things that has not been tested much, because it's one of those interesting groups of organism that creates a evolutionary link, as you know, between the prokaryotes and the eukaryotes, even though we thought they were the most ancient forms. So that's something that you might want to go through in detail once more and be much more aware of it. Also, there are areas about the classification, the taxonomic classification, about the kingdoms, the phylums and all that. So you might want to know the order of them. just in case they give you a very simple question to write them in a particular order. Another area, as I said, was the domains were not tested, and even the kingdoms, I mean, they have not been tested enough, I feel, in the Unit 4, where you need to know about the features of each of these kingdoms. So you might get a comparison over here, because even in the GCSE papers, there had been a lot of directing towards these kingdoms, the classification part and all that, which means the A-level syllabus also could be like that. Another area I feel for Unit 4 that could be tested is about the viruses, because we had the pandemic and all that, and enough time has passed by to ask you a question about the COVID virus and anything to do with the structure of the virus, right? So again, go through these sections, be familiar about its structure, how does it reproduce. Yeah, so that's basically what you need to go through there. Biodiversity, again, almost every year now you see a question on biodiversity, but I feel areas that has not been tested much is about to explain about the different levels of biodiversity. I know there was one recent question asking you what those levels are, but it was not much of an extended question, right? So you might want to go through these three parts as well. And the next thing you might want to go through is about the Link-Collins index of the distribution using the Bell transects and all that. But again, these are more practical oriented, so you might see them mostly in the Unit 5. But I think you still need to be thorough with the theory of that as well. Again, Spearman's rank correlation is more probable of coming in the Unit 5, but please do go through the theory behind it. Do a few questions on them as well, because there were a few Unit 4 papers that had these two, so you might want to still go through them. In terms of conservation, again, this is a hot topic, even in the Pearson syllabus, by the way. So you might want to go through the theory behind conservation. I would say go through all the things under conservation. But some of the things that I feel had not been tested much are these red areas about the role of the different conventions, like the IUCN, the CITES convention, like the good side of it, and even the bad side of it, because you probably would know that something like the CITES convention, I mean, it is done on a good intention, but the black market obviously increases because of this. So you might want to go through the pros and the cons behind these as well. And then, as highlighted over here, number 3 and number 4, which talks about the different conservation methods. Again, both the advantages and the disadvantages. And I also have a feeling that you might get to compare between two or more. Again, the good side and the bad side of them. So there had been a lot of questions, once more, as I said, on conservation. So this means that you need to look into more about that section as well. Coming towards the last part of this is about the genetic technology. Now, very interestingly, in the recent papers, there were lots of questions about PCR, and there were very interesting questions where the same question had something to do with PCR as well as gel electrophoresis as well. So that's really interesting, because you need to be able to relate them as in their use and all that. So you might want to be thorough with PCR as well as the gel electrophoresis section. I've not highlighted them over here, but then again, these areas that it more or less would come. Now, one of the things that was not tested very recently that much is your knowledge about microarrays, and this is a little bit of a challenging question, because most children do not know the use of microarrays. What is a microarray in the first place? So you might want to check some videos on them. You might want to go through your notes, because I feel that this is an area that has not been tested enough in the papers. So something to go through again. Just go back to some of these areas over here. So one of the other things that you would want to know is the process of genetic engineering, because one of the things that was asked very recently is how to create the DNA or the genetic material that you require for genetic engineering. Now, this is basically the same three that I've highlighted over here, but they have not asked a question about the vectors involved in the genetic engineering. They've also not asked about the different indicators that we use to identify if a particular DNA actually got modified. Right? This is where the fluorescent dyes and everything are being, fluorescent genes and everything are being used. So that's something that you might want to go through, because we normally tend not to go through it much. But again, because there was a question about how to create the DNA or the genetic material you require, this means this kind of information also you might want to go through then. Okay. Moving further is about the genetic technology applied, and very recently there was a question about SCID, and as I said, there was a question about Huntington's disease, and they basically asked about using genetic testing versus the genetic testing done in SCID, which one is better. And there was actually a comparison between why you can't do the Huntington's genetic disease until 18 years of age, but why SCID can be done a lot before that. So that was an interesting question where you need to think a lot about how to answer something like that. So you might want to go through things of this section, basically about how recombinant DNA technology can be used for insulin like diabetic treatment, then obviously identifying breast cancer and all that. And also to talk about always the ethical considerations behind them, because none of these are perfect. They could always give you a false positive or a false negative. And what are the other ethical concerns that are involved in these genetic technology? Now, an area I feel that has not been tested much is talking about how genetic modification helps in agriculture. Yes, there was a few questions, I won't say no to it, but this did not talk that much about the effect of GMOs, like the implications of GMOs in agriculture, how it helps to increase the nutritional benefit, how it helps in the crop yield. I think that's something that again was not tested enough in the recent papers. And again, the ethical and the social implications of using GMOs, because you know that there are certain people who still opt not to take products of GMOs, and you may have seen labels saying no GMO in the label itself. So this is another hot topic that you might want to go through, because we've not seen a question about GMOs for a very long time. Now, in the older textbook, I remember they used to talk about genetically modified salmon versus natural salmon, and there used to be a lot of past paper questions on how genetically modified salmons are beneficial for the fish industry, but at the same time how it is affecting the natural salmon, who are much more small in size. So this was not just about GMO affecting the human body, but this was actually talking about affecting biodiversity as well. So that kind of a question is something that you again might want to look at for this upcoming test. So I hope this video really helps you to think about what areas that you might want to start to go through actually. You might want to do a deep dive on this before the exam paper that is in another few days time. So consider subscribing to this channel. I'll be having more videos coming for the other unit papers as well. Leave a comment if you find this video helpful, and if you have any questions, please leave a comment as well. Thank you.